Assalamualaikum, my name is NURUL binti MOKTAR and our group will present about bioconcrete. First of all, concrete is an important and extensively used building material used for construction works. Concrete is the first option when building something to have a long useful life, which makes it one of the most used materials in construction. It is mainly used in heavy construction because of its strength and durability. Despite every attempt already used in the search for its improvement, it still presents considerable failure. No matter how carefully the concrete is mixed or reinforced, it eventually ends up in cracking by expansion and contraction at some point, which result in an unattractive appearance and can also compromise the entire structure of the building. Crack also increase the permeability of matrix, thereby increase the chance of corrosion in reinforcement. Large crack may affect the structural integrity while the small crack reduce the durability of the structure. Therefore, the sole factor of structural failure is cracking. In order to reduce the chance of crack formation, a structure requires regular maintenance that can be costly and may further increase the maintenance cost of the structure. To solve the problem of crack in concrete structure, researcher Hendrik Marius Jonkers observed that the human body, including the bone structure, can heal minor damage on its own. Through this inspiration, Dutch microbiologists have developed a bio-based, safe regenerating concrete that can overcome the crack and also increase durability of structure. The use of this new material in new construction and maintenance of existing structure will substantially reduce the cost of the preserving of concrete structure. The bacterial cells are potential admixtures in concrete, help in enhancing the mechanical performance of concrete. So, what are the properties of bioconcrete? According to Kumar et al. In 2019 in his paper entitled Properties of Bioconcrete, the workability test of bacterial concrete resulted in 90 mm of strength value. This is shown that bioconcrete has a high workability. The biological treatment of the cement composite results in the crack sealing. Therefore, bioconcrete has the ability to heal the microcracks with the help of bacteria and healing agent. Next, bioconcrete has low permeability, as water could not flow through the microcracks. Next, bioconcrete has a high compressive strength. After 91 days, the compressive strength of bioconcrete has reached to maximum strength. Compared to the conventional concrete, the bio-based cement composite has the higher durability of concrete structure as its mechanical properties is improved. Additionally, self-healing concrete also help in reducing the maintenance and repair costs.
Basic concept will be covering 4 components, which are type of bacteria, testing method, application, and factor affecting. Type of bacteria consists of anaerobic bacteria and aerobic bacteria. Example of aerobic bacteria is Bacillus pasteur, Bacillus cephyricus, Escherichia coli, Bacillus cornei, Bacillus subtilis. The testing method for the bioconcrete consists of 3, which are flexural test, compressive strength test, and speed tensile strength test. Application of bioconcrete can be applied to existing building in form of spray, prevent reinforcement from corrosion, reduce permeability in concrete, and bring great advantage to marine and submerged structure. Factor affecting for bioconcrete consists of 4 factors. The first one is size of crack, pH of concrete and crack, temperature, pressure and healing time, and the presence of water and air. For the size of crack, it is very difficult if the size is too big. Next, for the age of concrete and crack, the effectiveness of self-healing will be decreases as the age increases. Next is temperature, pressure and healing time. The healing improve with higher pressure and temperature. Lastly, the presence of water and air. It improves that the healing process will be better with the presence of water and moisture content.
Hi, my name is MOHAMAD HAFIZ. I would like to share about the material used in this case study. First there is ordinary Portland cement, natural sand, asphalt aggregate, crushed stone as coarse aggregate, and water. For the microbial material, there are 3 types, which are Bacillus pasteur, Bacillus cephyricus, and Bacillus subtilis. For preparation of bacterial solution, the first preparation is mixing. In a conical flask, 50 ml of distilled water was mixed with 1.25 grams of nutrient broth. The second preparation is sealing. To avoid contamination, the conical flask and medium were sealed with cotton balls and wrapped with silver foil. For the third preparation is sterilization. For 20 minutes, the medium was autoclaved at 121 degrees Celsius and 50 pound pressure. For the fourth preparation is inoculation. The pure culture of different selected bacteria was inoculated into the 50 ml of nutrient broth solution under aseptic conditions within UV sterile laminar and flow chamber. and the final preparation is incubation. The samples were placed in an orbital shaker and were incubated at 37 degrees Celsius and 125 rotation per minute for 16 till 18 hours. For the test conducted, concrete mix was prepared by using ratio of M20 concrete, which is 1, 1.5 and 3. Hence, for every 1 part of cement, 1.5 parts of sand and 3 parts of aggregate in volume were used to prepare M20 grade concrete. Next, Bacillus subtilis was added to the concrete at a proportion of 105 cells per milliliter. Next, the same proportions are used in all bacterial concrete. Next, all the steps were repeated by using different types of selected bacteria. And the following tests are conducted on these specimens to determine the strength of concrete, which are compressive strength, split tensile strength, and flexural strength. Compressive strength result. Table 1 shown that comparison between compressive strength of bacterial concrete and conventional concrete. 3 microbial materials were tested, which are Bacillus subtilis, Bacillus sphaericus, and Bacillus pasteurii, along with the conventional concrete. The result shown that Bacillus sphaericus have the highest compressive strength during 7 days and 28 days of curing, which are 34.58 Newton per millimeter square and 45.72 Newton per millimeter square respectively. Split tensile strength result. Table 2 shown that comparison between split tensile strength of bacterial concrete and conventional concrete. 2 microbial materials were tested, which are Bacillus subtilis and Bacillus sphaericus, along with conventional concrete. The results showed that Bacillus subtilis have the highest split tensile strength during 7 days and 28 days of curing, which are 2.36 Newton per millimeter square and 3.73 Newton per millimeter square respectively. Flexural strength result. Table 3 shown that comparison between flexural strength of bacterial concrete and conventional concrete. 1 microbial material were tested, which are Bacillus pasteurii, along with conventional concrete. The results shown that Bacillus pasteurii have the highest flexural strength during 7 days and 28 days of curing, which are 4.6 Newton per millimeter square and 7.85 Newton per millimeter square, respectively.
The compressive strength was assessed for 7 and 28 days of curing concrete specimen. The target mean strength was reached for M20 grade concrete for conventional concrete, which is 29.99 MPa. There is an improvement in compressive strength for all type bacteria used compared to the normal concrete. However, the result shows by using bacteria of Bacillus sphaericus obtained the highest value compared to others bacteria, which 38.58 MPa at day 7 and 45.72 MPa at day 28. The compressive strength was regained up to 52.42% higher at day 28 compared to normal concrete due to settlement of calcites as filling material in inner portion of the cracks concrete surface. Similar to the compressive strength, the split tensile strength was computed for 7 and 28 days of curing. There is a rate of 38.17% of split tensile strength for bacterial sample of Bacillus subtilis after cure for 7 days and 14.14% for 28 days compared to the normal concrete. It is because the formation of the EPS layer by the bacterial strain. The split tensile strength for day 28 increased to 3.73 MPa from the 3.26 MPa after cracks healing. There is a regain of flexural strength of 7.85 MPa for bacterial mixed specimen of Bacillus subtilis from 7.06 MPa ordinary concrete sample after 28 days. There is an increase of 11.18% of flexural strength after 28 days curing by the excretion of urease enzyme of biomaterial. Sorptivity test is an indirect measure of permeability of concrete. It is to monitor water penetration resistance of the concrete. It resulted that the bioconcrete offers more resistance against passage of water into concrete, hence the porosity decrease. The precipitates produced by the bacteria in the pores of the concrete improve the denseness of the concrete specimens that leads to the decreases in sorptivity values.
From the study, we can conclude that the bioconcrete is a concrete which can healing itself from minor crack, where it can reduce the maintenance cost and increase the age of the concrete. The presence of water and air will help to heal for the healing process more effectively. From the study, it shows that the strength of the bioconcrete is higher compared to the normal concrete in the form of compressive strength, split tensile strength, and flexural strength. That's all from us. Thank you.